We're going to switch gears now. That last talk was obviously theoretical. We're going to switch to an experimentalist. Our next speaker is Professor David Chenoweth. David is the youngest member of his group. He's only been at Penn for a few years, but he's already made a name for himself in C&E News by chemically modifying collagen to make it much stronger. I don't know if he's going to talk about that today, but we'll see. But anyway, David Chenoweth is in our chemistry department, and again, he's works on biological chemistry theory and research. Not theory, but research. So David.
Okay, thanks for the introduction, Drew, and thanks for the invitation to talk here. It's a pleasure to be here. It's my first time in Puerto Rico. Puerto Rico's beautiful. I definitely need to come back soon with the whole family. So today, I'm going to tell you a little bit about one of the most abundant proteins in the animal kingdom, collagen. It's a really interesting biopolymer. It serves a structural role, but there's also many roles that it serves beyond just structure. So our research group, we're interested in building small molecules and making synthetic modifications to biopolymers to modify their properties. And to use these, we also use these small molecules in these modified biopolymers as probes for different cellular environments. So today I'm going to focus on collagen, which the structure of the most fundamental structure, the triple helical structure, shown right here. And so collagen, it's one of the most important biomaterials in the animal kingdom. It occurs in tendons and ligaments, skin, bone, hair, teeth, our fingernails, and it has very unique properties. So you can imagine just the properties of bone versus the properties of skin. They're so different. So collagen can serve a structural role in skin, but it can also serve a role in bone where it forms a composite, solid state material and gives strength to bone. It forms it's a major component of the extracellular matrix. So the environment that the cells interact with outside, the basement membrane, it's used as a biomaterial. It's used for bone grafts, cosmetic surgery, tissue regeneration, wound care. It's used in materials that are used to grow stem cells, matrigel, and mutations in collagen. In the amino acid sequence of collagen, it forms the basis of many human diseases. Here's one in particular, osteogenesis imperfecta, where it's just a single amino acid mutation that causes this disease. And this is sometimes called brittle bone disease. So the backbone of collagen, or the repeating sequence, it's often this proline, hydroxyproline, glycine repeating sequence. So it's a biopolymer that's formed by triplet repeats. Every third amino acid is almost always glycine in this case. So mutations in glycine, it can lead to a lot of these human diseases. Glycine is the one amino acid that really can't be switched. It's strictly conserved in collagen. There's a lot of early work that went into determining the structure of collagen. This triple helical structure that you see right here, this is from a X-ray crystal structure. But early on, Linus Pauling worked on this problem. Ramachandran, Francis Crick, Rich, Ada Yonath, Brodsky, Berman, all of these greats in structural biology worked to figure out what the higher order structure of collagen was and how these peptides self-assemble into this structure. And then there's been many synthetic modifications throughout the years, which have been, the goals of these studies have been aimed at modifying this backbone sequence and trying to enhance the properties of this biomaterial. And there's been a lot of beautiful work there from some of these groups listed here. This is a notebook page from one of Linus Pauling's notebooks. This is 1951. It's a little hard to read here. But back in the 50s, they didn't know the structure of collagen. So Pauling, when he was doing all this speculative model building at the dawn of molecular and structural biology, he was trying to figure out what the higher order structure was. And he got the triple helix part right. He actually figured out in 1951 that 3 strands should come together and form a triple helix like this. And then he was, with just pencil and paper, he was trying to work out how these 3 peptide strands recognized each other and how they fit together. And so he just started drawing models of the peptide and how they might interact. And at that time, he was kind of enamored with hydrogen bonding. And so he was trying to figure out how you could maximize hydrogen bonding from the NH of one amide on one of the peptide chains to the carbonyl on the next peptide chain. And so he drew this structure out here and tried to maximize hydrogen bonding between the three strands. And if you notice here, he thought hydrogen bonding obviously was very important. So he's maximizing hydrogen bonding in this picture at the expense of putting in cis-amide bonds. And so we know now that cis-amide bonds are not tolerated in collagen. And the structure really doesn't have cis-amide bonds. Pauling got the structure wrong, and some other people figured it out later. But that's okay. This idea of maximizing hydrogen bonds, it turns out that's actually really important. And that gave us an idea for the project that we started working on in my group when we started. If you look at collagen sequence alignments, so here's just looking at a particular type of collagen, human, mouse, chicken, dog. So if you look at all these, you'll see how strictly conserved glycine is. So in this dark kind of purple color here, that's glycine. And so in about every third position, you see glycine repeated over and over and over again. And mutations in that will lead to disease. Collagen also, it doesn't just form it doesn't just serve a structural basis in biology, but it also serves as a scaffold for recognition with other proteins. Recognition with proteins inside cells and outside cells in the extracellular matrix. And so here's just a few crystal structures that we currently have of proteins as they're interacting with the collagen triple helix. And so this in particular right here, matrix metalloprotein 1, this is a protein that processes collagen and dynamically reorganizes collagen. So these are really important, especially in cancer biology, where these are thought of as a target for therapeutics for cancer biology. It also serves as a scaffold for multiple proteins to assemble onto collagen. And some of these sequences, one of the most popular sequences for protein recognition is this proline arginine glycine. So here you can see now we've switched out the hydroxyproline for an arginine. And that side chain will make molecular recognition interactions with some of the proteins that interact here. And so this is where, so I'm going to tell you about a project from one of my first graduate students. This is Yitao Zhang. He's a fifth-year graduate student. And his project was to try to figure out how to enhance the properties of collagen so that we could make more stable collagen fibers, so that we could minimize the collagen peptide motif, make a very short peptide that would still self-assemble into a triple helical form and recapitulate the properties of collagen. And so a lot of work had been done to modify the side chains of collagen. So to take this proline, for instance, and put some other substituent on this proline to enhance the stability, or to mutate out this hydroxyproline and put an unnatural amino acid there. The problem with that is you can get a little bit of stability, but you change the overall surface topology. So the way that biology sees collagen then is different from the natural sequences. And so we wanted a way to enhance the material properties of collagen, but not change the overall surface topology of collagen. And so we focused in on this glycine position here and the alpha carbons in the backbone. And it turns out no one had ever modified these alpha carbons in the backbone of the amino acid chain. So what we did to start is Yitao made a standard collagen peptide. So this is a repeat. It's a 21-mer peptide of proline, hydroxyproline, glycine. So that repeats 7 times. If you build that peptide and you just let it stand in solution, it will spontaneously self-assemble into this triple helical structure here. And then what you can do is you can take a CD spectrum of that, and here's the CD signature of that triple helical peptide. You can focus in on any wavelength along here, and you can heat it up. And when you heat it up, you can map out an isotherm for the melting of that triple helix structure. So you can unfold the triple helix, and you can refold the triple helix. And then at this 50% mark here in the unfolding or the inflection point, that's the TM, or the thermal unfolding temperature. And so that's going to be an important parameter for us to follow when we're assessing different mutations to this collagen peptide and how they affect the stability of the structure. So that's one technique we can use. Another technique we can use is we can completely unfold that triple helix. We can take it to one temperature, hold it at that temperature, and then we can watch the kinetic refolding as it spontaneously refolds and self-assembles into a triple helix. So that's another thing. That's another experiment we can use to assess it. And then we can prove that it actually is a triple helix by standard methods like size exclusion chromatography coupled with multi-angle light scattering, and we can use analytical ultracentrifugation to show that it's actually in a triple helical form. So these are just standard biochemical, biophysical methods that we can use to study the state of it and some of the properties. So when Yitao started his PhD, they had 3 simple experiments to do. And the 3 simple experiments were, let's replace the alpha carbon of glycine, which has never been able to be replaced. Let's replace the alpha carbon of proline in the X position, and then in the Y position here, let's replace that alpha carbon. And so here, if you took this alpha carbon now and replaced it with a nitrogen and modeled that into one of the collagen crystal structures, what we noticed is it should form an extra hydrogen bond. And that extra hydrogen bond would be at the interior of the collagen triple helix, not on the exterior. And so that shouldn't change the overall topology of the collagen triple helix. It should just maybe, if we're lucky, add a little bit of stability. Now if we move that over to this position, here you notice we're actually replacing a stereocenter with a nitrogen. And so we've got a configurationally stable stereocenter in proline, and we're going to try to replace that with nitrogen. You'd think that might actually be bad. But if you model that into the crystal structure, it turns out nitrogens can adopt a pyramidalized form, and a pyramidalized form which looks like a tetrahedral form of a carbon stereocenter. It's just the hydrogen that used to be here on the carbon is now, that position is now occupied by a lone pair. And in this position, that lone pair would be solvent exposed. Now if we move that over 1, the same mutation here, the same carbon for nitrogen substitution, puts a lone pair, it projects a lone pair right into the center of the collagen triple helix. Now that, in that position, it looks like there's a lone pair interaction with this carbonyl here. And so the prediction was, this should be stabilizing. This should be maybe stabilizing, maybe destabilizing. We didn't really know what was going to happen there. This we predicted to be destabilizing. And so one of the first experiments that Yitao did is he made these 2 replacements here of this proline for azaproline and this proline in this position. And what he found is that indeed a nitrogen can actually mimic a stereocenter in this position. And so this line here represents the unfolding or the TM value for a standard proline hydroxyproline glycine collagen triple helix. So with 1 modification of a carbon for nitrogen here at that stereocenter, we get the same unfolding profile at this temperature. So it looks like it doesn't affect the triple helix stability. And then, as we suspected, if you put it here in this position, there is a lone pair interaction. We just didn't know what it would be worth. It turns out it's worth a lot, and it doesn't even fold and assemble into a triple helix. And then the surprise came when we modified the glycine. And so when you modify the glycine, when you replace just a single carbon for nitrogen, the thought was that might add an extra hydrogen bond. And it turns out it looks like it does. So we get about an 11-degree increase in the stability of the triple helix when we do that. And so it's a single atom substitution in this peptide here. So 1 atom out of about 240 atoms. And you get this much stability out of that. You get 10 degrees. So that we thought was pretty interesting. And then we decided, okay, what happens if you put 2 in? So how about a 2-atom change? So with a 2-atom change, it's additive. You get almost 20 degrees. And so now here's 2 atoms next to each other, or here's 2 triplet repeats next to each other, and each glycine has been replaced by this azoglycine that contains a nitrogen for carbon substitution. So now what happens if they're not next to each other? but you skip space them. And if you do that, it turns out it costs you a little bit in thermal unfolding. And so you get a little bit of a decrease in the melting temperature of the triple helix when these are now skip spaced. And there's a normal proline, hydroxyproline, glycine triplet in between these two. So then if 2 is good, what about 3? So now 3 atom substitution. So 3 carbons are now replaced with a nitrogen, and you go up another, slightly over 10 degrees. So with 3 atoms, now we get a 34-degree increase in the thermal unfolding of this triple helix. And now what about skip spacing? So these are all adjacent, right? So what if we skip space all of them? So if you skip space them, again, we see that there's a cost for that skip spacing. So now when you skip space, you decrease, it's a little bit less stable. And so there's some synergism of having all of these azoglycines in the triplet repeats that are right next to each other. So now if you add just one more triplet repeat, it turns out the thermal unfolding temperature is so high we can't measure it. You're getting close to the boiling point of water. And so at this point we decided, okay, you've got a 21-mer peptide. Let's see how short we can make it. So it turns out a 21-mer peptide was the smallest peptide sequence that could ever be assembled into a triple helix. So what if we just chop it down? How short can we go? And so here we've now chopped it down to just 12 residues. So here's 12 amino acid residues. 4 of the glycines are replaced with azoglycine, so it's a 4-atom change. And now we have a triple helix that unfolds at about the same temperature as a normal 21-mer of collagen. So we can almost cut it in half and just change the 4 atoms and get the same stability. So now we really have minimal collagen peptide motifs that act like they spontaneously self-assemble like triple helical collagen or longer. And then if we add just one more triplet, so one more atom, one more glycine change. So now this is a 15 residue peptide, and we look at the unfolding of that from the triple helical form. Now we're up here at slightly over 80 degrees, but still a very short peptide. And then if we look at positional scanning, so we ask the question, does it matter if the azoglycine mutation is in the middle versus here versus at the end? So if we just take that azoglycine, a single atom substitution, and we scan all the way through, we see that in the middle, it's the most stable. It gives you an 11-degree stabilization. But as you go to the end, the very end here, it's still worth 6 degrees, which is pretty interesting because there's a phase shift in the 3 strands by 1 amino acid. And so at most, there could only be 1 or 2 extra hydrogen bonds at the end, not 3 total. And the ends are fraying, too, constantly. So it looks like it's stabilizing no matter where you put the substitution. And then if you look at multiple substitutions, here's the adding 2 azoglycines that are spaced far apart. That's worth about 17 degrees. And then as you bring them closer together, 19, and adjacent to each other, 21. And here's the skip spaced, that's 29, but as you bring them very close together and they're all adjacent, that's worth 34. So there's some synergism there. And then if you look at the short sequences, here's a 9-mer now. So the 9-mer doesn't self-assemble, it's too short. So when we get to 12, that seems to be the critical length for getting self-assembly. And so the thermal unfolding is around close to 40 degrees. And then with one more, it's up around 78 degrees here. You can also look at the refolding. And so how fast do these spontaneously refold into a triple helical form? So here's our standard proline, hydroxyproline, glycine repeating 21-mer. So that refolds with a T1/2 of about 24 minutes, and that's this blue curve right here. And now, when you look at these azoglycine-containing peptides, it turns out they refold much faster. So the kinetic refolding times are much faster. And if you just put those all on a plot and compare them, so this blue dashed line is that standard proline, hydroxyproline, glycine repeat 21-mer. That's our baseline there. Here's our single atom substitutions as we scan along the backbone. So there's a maximum in the middle here where we put it dead center on the 21-mer. Here's now 2 And as we bring two closer together, when they're next to each other, when they're in adjacent triplets, it's worth the most. And when they're spaced farther apart, there's a little bit of cost for doing that. With 3, then you bump up again here. So 3 atom substitution puts you here. And then when you chop the triple helix, or you chop the peptide down to a minimal motif, with a 12 amino acid motif, you can get a peptide that unfolds somewhere in this range, and with 15 up here. So one of the previous records for a synthetically modified collagen peptide was this one right here, where 14 of the prolines had been replaced with a methylproline and fluoroproline. So if you replace 14 of those in a 21-mer, that would be this line right here. And so as opposed to replacing 14 amino acids, we can just replace 2 atoms and get a collagen peptide that's just as stable. But it doesn't change the overall topology on the outside of collagen. And so then if you compare that to just natural repeats that self-assemble, here's a glycine proline hydroxyproline repeating structure. When you get up to 45 amino acids, and so this is 15 repeats of this triplet, you're right about a little above 80 degrees. And so now we can do that with 15 amino acids instead of 45 amino acids. And now we're thinking about, so those were glycine they were proline hydroxyproline glycine repeats. So what about other sequences? We're wondering if this is a general effect. So can we use azoglycine to stabilize other sequences of collagen that might be important for protein-protein interactions? And so one really common motif is this proline arginine glycine motif here. And this is important for interaction with a protein called HSP47, which is a really hot cancer target right now. So we looked at this modification right here. We wondered if we replaced a collagen peptide, the glycine, next to an arginine, if that would still be stabilizing. And so here is a standard collagen peptide that we made. Again, it's got 3 repeats of this proline hydroxyproline glycine, and then embedded in the middle is that recognition sequence for proteins, for especially HSP47, where there's an arginine now here in this position. And then glycine, and then another 4 repeats of proline hydroxyproline glycine. And that has an unfolding curve that looks like this right here. And so cooperative unfolding that's just a little bit above 40 degrees. And then if we just take that one atom right there, that carbon, and we replace it for a nitrogen, here's the new thermal unfolding. So we gain about 10 degrees in thermal stability just by making that one atom substitution. And then if we look at refolding, it turns out it refolds much faster again. And we've done some molecular dynamics calculations to look at this and try to figure out if it really is a hydrogen bond at the interface. And so here's an MD simulation just over 20 nanoseconds where now you're looking at 2 standard collagen peptides, and then you're replacing these glycines with azoglycine. And a really interesting thing that happens here is when we look at the molecular dynamics simulations, it looks like the RMS deviations across this triplet, we're getting larger fluctuations. And so we're increasing the dynamics in that region. We didn't actually know why. We expected it to be more stable, not more dynamic with this extra hydrogen bond. And so what we think is happening, when we look at the MD simulations, it turns out this extra NH that we're adding in place of that carbon, it's choosing between 2 different carbonyls, and it can flip back and forth, and it can form a hydrogen bond to this one, or it can form a hydrogen bond to this one, or it can form a bifurcated hydrogen bond. And as you heat that up, you could imagine it's a desolvated interface. And so in this material, it's kind of dynamically reforming hydrogen bonds and kind of holding it together. So that's what we think's going on from MD simulations. And if you plot out the angles and the distances for all these hydrogen bonds, it turns out they fall right within standard hydrogen bonding parameters. So we were also recently able to get a high-resolution crystal structure of one of these. And so now here's that collagen peptide that I showed you, where we have the arginine here, which is important for recognition of different proteins. And now it has that extra nitrogen replacing a carbon in this position. So we were able to take this peptide here and crystallize it to 1.1 angstrom resolution. And this is the triple helical structure shown here. This is how it packs. And this is the solvation right here. So all of these aqua spheres, that's the solvated triple helix. So you can see it's packed. in this arrangement, and then when you solvate it, you can kind of fill in all these gaps with all the water molecules. So this is, let me just start this movie here. So here's a movie of the electron density map. So as this zooms in, you'll see the hydration state. Here's all the waters around the outside. It's really well ordered. And what I've done here is where the alpha carbon used to be, it's now nitrogen, and I've colored that in dark blue. So now you can see you get an extra hydrogen bond there, an extra hydrogen bond there, and an extra hydrogen bond there. And if you take a slice through the triple helix, you can see here's where glycine used to be, and now this is our azoglycine modified amino acid, and that's that extra nitrogen, and it forms a hydrogen bond with that carbonyl there. And so in the crystal structure, we only see one of those. We don't actually see it flipping back and forth between the two carbonyls. So we're not sure if this, if what we see in simulation is real or an artifact of the force field or not. But in our crystal structure, we see this. And if you plot out all the hydrogen bonding parameters, so here's the angle from the oxygen of the carbonyl to the hydrogen to the nitrogen. And these are pretty optimal hydrogen bond angles here and hydrogen bond distances here. All the extra hydrogen bonds we're adding, they're represented by this little pink sphere or little pink circle here, and those all fall within standard hydrogen bonding parameters. And then here's the distances if you actually put the proton back in and look at those, and those are all short hydrogen bonds. And so now we're interested in using these and looking at do they really interact with biology in a normal way, or does the overall topology get changed? And so one really interesting experiment is to think about, well, how do these interact with matrix metalloproteins and other proteases or collagenases that will actually degrade collagen? So we ask the question, will these be degraded by some of these proteases and collagenases? And so here's the next experiment we did, where we took just a standard collagen peptide and exposed it to collagenase enzymes that will normally break it down. And then we looked at how fast it'll break down normal collagen versus how fast it'll break down one of our modified collagens now. And so you can buy collagenase enzymes. They're used for tissue dissociation and cell isolation. They're also used as cancer therapy to degrade the extracellular matrix around tumors. And so we bought a standard collagenase. We looked at some different concentrations in the CD spectrometer, and you get minimal signal at this concentration. And then we added our collagen peptide, our model system collagen peptide. And here's the triple helical form. You can see that over time, that gets degraded. And so here's the degradation of that triple helical form over time when you expose it to a certain concentration of these collagenases. And then we looked at one of our azoglycine-containing collagens and compared it to that. And so when you do the comparison, it turns out the collagenases, they don't touch this when it has azoglycine in here. And I'll just skip past a couple of these things here. So it turns out after exposure to collagenase enzymes, you can't refold this one. You don't get a cooperative refolding curve, but you can refold this one still, so it's not being degraded. And then, I'll just move on here. We also look to see if we add our synthetically modified collagen to a standard collagen, would it inhibit the activity of the enzyme? And so is it really being recognized by the enzyme that's degrading these? Or is it just being ignored? And it turns out it's actually being recognized, and it actually acts as a potent inhibitor. And so as you increase the concentration of this in the presence of standard collagen, it inhibits the degradation of standard collagen. And so now we're thinking we can probably make sequence-specific inhibitors of matrix metalloproteins and other collagenase enzymes with this modification. And we're also thinking about biomaterials applications, and so if you just plot out Young's modulus versus strength of all these different proteinaceous biomaterials, collagen falls right here in the center. So if you think about adding in azoglycine, it's interesting to think about where can you shift the properties of normal collagen to on this plot. I'll just go to the most important slide. So these are the students who worked on this collagen project, Yitao Zhang, and this fantastic master student in my lab, Alex Kasnell, who's done all the crystallography work. Roy Malakamal, and these funding agencies, and I'd like to thank you. And if you have any questions, I'm happy to take them.
Fascinating presentation. Any questions?
Hi, really nice. So, I mean, there's the potential to use these as therapeutics, I guess, in people with collagen problems. So you answered part of my question at the end, showing you can interact these hyper-stabilized collagens with normal collagen. Do you anticipate delivering these very small molecules as therapeutics, or do you give them as RNA and then have them be translated? Or?
That's a good question. We don't.
Could you repeat the question for the people
Oh,
in the back, because I'm sure we can't hear.
Sure. So do we anticipate using these as therapeutics? And if so, could we deliver these, and how would we deliver them? What we're thinking about now is we're thinking about you could make small molecules that would be inhibitors that then are on the order of the molecular weight of certain drugs and have different properties of drugs. But you could also think about making many proteins that interact, that are protein-protein interaction inhibitors. And those might be useful in the extracellular matrix, or they might be useful intracellular also. There's also a lot of host pathogen interactions where pathogens, parasites, viruses, things like this will excrete proteins that will then degrade the extracellular matrix. And so you could think about small molecules or mini proteins made of collagen peptides like this that would inhibit those interactions. So
people with osteogenesis imperfecta, right?
Yeah.
You're essentially wanting to supplement.
For those type of genetic diseases, I don't know that this would be useful. You'd have to I think that'd be tough. Yeah. I think more than anything, these are going to be useful for new synthetic materials, probes for structural biology, and we're also thinking about imaging agents where you could use them to image the extracellular matrix and putting super resolution fluorophores on, having them integrate into collagen defect sites, and then being able to image processes and things like that. But correcting some of these genetic diseases, I think would be tough.
Beautiful work.
Yeah, thanks.
So just I want to make a comment just for the students' benefit, right? So this is a sample process. The T melting depends on the concentration. Right?
Absolutely.
Because so now you do not determine
Absolutely.
exactly the room concentration.
Yes.
The question is, how do you determine the concentration accurately?
Right. So we make enough of it so that we can actually weigh it out and determine accurate extinction coefficients and all of these things. Yeah. So you have to. So there's standard extinction coefficients that you can use to calculate the extinction coefficient of a peptide, and then you can use UV quantitation to do that. And so we have to establish similar things for collagen peptides that have this modification in them.
Okay, so my question is that it looks like there are cooperativity, right? So between the units, right, even they're further apart. Have you tried to use the simple Ising model to tease out a little bit more?
Not yet.
Energetics?
Not yet, but we definitely need to.
Yeah, perfect.
Yeah, and so you can do the unfolding and the refolding, and you can look at the hysteresis in that, and you can extract thermodynamic parameters from that. But the problem is you're changing temperature, right? And you can't assume that the heat capacity is constant the whole way through. And so we'd really love to do differential scanning calorimetry and get really good thermodynamic parameters. But we don't have a DSC, and we haven't done that yet. But we'd love to do that type of thing.
So I'm going to ask my first question. Do you see that increase in cooperativity as you go to longer chain lengths? Do you see a sharp step-like transition?
That's a good question. We haven't went to very long. Yeah, we haven't went much longer than what I showed you there.
So you're saying that it's a relatively short.
Yeah, as you get longer, the stability gets really high really fast.
Okay.
And then you could just boil water and they don't come apart.
Okay, and then the second question I had was the last part of your talk. You talked about using these as inhibitors. Can you, from your enzyme kinetics, can you pull out a KI?
Yeah, so we haven't done that yet. We need to do that, but we think it's pretty high. I mean, yeah, that steep transition is over the course of about 0.1 equivalents, and we haven't done that yet, but we will. Okay.
No more questions. Thank you.